Are cells immortal? In the sense that they can divide with an unlimited number of potential offspring, some cells are. Obviously, bacterial species, amoeba species, etc., continue generation after generation. There is no potential end to their cell divisions. In the human body, this is true to an extent because the human species can continue after generation and generation, and cells are obviously dividing to produce new human individuals. And so in our germ tissues, in the gonads, and embryonic stem cells are potentially capable of an unlimited number of cell divisions. So these cell lines will continue, perhaps without end. Most of the cells of the adult human body, however, are not immortal in this sense because they only have a limited number of cell divisions that they are capable of. A major reason for this is that they typically turn off the enzyme telomerase. They no longer produce it. This enzyme helps to lengthen the ends of chromosomes during cell division, without which chromosomes become shorter every time a cell divides, and thus a cell would only have a certain number of cell divisions that it would be capable of before the chromosomes would simply be too short to allow additional cell divisions. This is one of the major reasons for aging. Individuals may get to a point later in life where cells have a limited ability to reproduce because many cells have simply shortened their chromosomes to the point where they can no longer divide. Cancer cells are a threat when they mutate and continue to express telomerase when they should not. This gives them an potentially unlimited number of cell divisions. They are potentially immortal, these cell lines. They can divide and divide without end. While this is a great potential risk to the human body, it is a valuable tool in research because cancer cells can thus be cultivated in cell culture without end. These cell lines are potentially immortal. If one had a hybridoma, which was producing a desired type of antibody, well, this would be something that one could potentially produce indefinitely because this cell lineage could be replicated and replicated through tissue culture, and the cell line would be immortal because it was producing telomerase and its chromosomes were not shortening with each cell division.